Thank you very much for the introduction. It's a great pleasure to be here. And before I start my talk, I'd like to acknowledge the contribution of my colleagues from Carnegie Mellon University, my advisor Carl Olson, and also my colleagues at Janssen Pharmaceuticals and the Inscopics. Without these people, none of the projects I'm going to talk about would have been possible. So the goal of today's talk is to introduce you to the general logic of the experimental approach to investigate neuronal mechanisms of normal and abnormal behavior. For the goals of today's presentation, I'm going to define neuronal mechanisms as discoverable and causal relationship between activity of neurons and behavior. And I will start out by describing two most commonly used nowadays approaches to record activity of large sets of neurons in awake behaving animals. I will exemplify each of these approaches by describing a particular project we've completed. In the first project, we used multiple electrodes to investigate neuronal mechanisms of serial order behavior. And in the second project, we were studying neuronal mechanisms of behavior changed by a drug. But let's start with describing most commonly used approaches to record activity of multiple neurons in awake behaving animals. To link behavioral and neuronal activity, of course, we need two components. We need to monitor in the set of carefully controlled experimental conditions the behavior of the animal. And at the same time, we have to record activity of neurons in the animal's brain. There are multiple ways of recording activity of large groups of neurons. However, most commonly used approaches nowadays are the use of multiple electrode arrays, and this technique relies on the measuring voltage changes near the tip of the electrode, and these voltage changes are associated with the action potential fired by a neuron. Calcium imaging, on the other hand, relies on the phenomenon that is very highly reproducible, and its rise in the intracellular calcium which accompanies action potential. And this rise in the intracellular calcium can be measured by the use of fluorescent calcium indicator. Two techniques are comparable at the moment as used in everyday lab in the number of neurons you can study at the same time. However, it seems that the calcium imaging has the higher upper bound on the number of neurons you can record. For example, it's been recently demonstrated that you can record every neuron in a brain of vertebrate animal. Both techniques allow distinguishing across neuronal types. However, in case of calcium imaging, you can specifically target the expression of calcium indicator in genetically identified neuronal population. With the use of multiple electrode arrays, you can distinguish neuronal type by the by the characteristic shape of the actional potential. However, you do not know about the genetic makeup of this particular cell. The multiple electrode arrays are often constrained by the array's geometry. Therefore, you can record neuronal activity only at the very tip of each electrode. Calcium imaging allows studying more natural arrangement of neurons in the visual field. However, the flip side of this is that often to expose the optical field, you have to make the cranial window and oftentimes remove the overlaying tissue to expose the area of the interest. The multiple electrode arrays are somewhat less invasive in this respect. And finally, the advantage of electrode arrays is the superb temporal resolution, microsecond scale, which allows you to study individual spikes fired by neurons. Calcium imaging is often constrained by the dynamics and kinetics of the particular calcium indicator in use, and this temporal resolution in the order of milliseconds. Therefore, it makes it more difficult to resolve individual spikes. However, there is a set of molecular tool which became recently available, which actually allows you to do that. However, it's not well validated yet. So let's start by demonstrating the use of multiple electrodes to study neuronal mechanisms of serial order behavior. What is the serial order behavior? We know that in everyday life, we're often performing actions in the particular serial order. For example, the series of dance steps. We're also capable of performing and selecting objects in a particular serial order. For example, when we add one by one ingredients while cooking. Presumably, Our ability to appreciate and understand serial order relies on the activity of certain neurons somewhere in the brain which can signal ordinal position. And the first question we asked in this project, are there neurons which in fact can signal an ordinal position within a sequence? In other words, do rank selective neurons exist? And the second question, if such neurons exist, are they capable of representing serial order abstractly, regardless of particular behavioral context? In other words, would the neuron which signal the second position in the sequence of action would also signal the second object while you're selecting the objects in the particular order? How abstract this representation of rank is. To address this question and general issue, we recorded in 4 areas of monkey frontal cortex. And we recorded activity of multiple neurons while the monkey was performing serial order task. The first question we asked, do neurons exhibit rank selectivity in the context of an action sequence task? To answer this question, we designed a task, which I demonstrate next. The trial began with the central fixation. The monkey was required to look straight. Then 3 identical pictures appeared on the screen. The identity of pictures signaled to the monkey in which order he was supposed to look at them. For example, the picture of the sailboat indicated that the first movement should be to the left. If the monkey executed the movement correctly, the feedback appeared on the screen, and then the monkey returned to the central fixation. In the second phase of the trial, the correct movement was upward, and in the third phase of the trial, it was to the right. If the monkey performed the entire sequence of actions correctly, left, up, and right, at the end of the trial, he received the reward, a drop of juice. In the sequence I've just shown, the correct order of ocular movements was left, up, and right. However, we trained the monkey to perform 6 different sequences in which the direction and rank was counterbalanced. We found that in the context of this particular task, neurons indeed signaled rank. Let me give you an example. In the example sequence shown before, left, up, and right, this particular neuron fired action potential indicated as points here on the raster plot during the phase 1 of the trial. But how do we know? Does this neuron signal rank or direction? We know that because we recorded the activity of the same neuron across 6 different sequences. And now, by separating neuronal activity by direction and rank, we can clearly visualize and confirm that, in fact, this neuron was selective for the rank 1. We also found the examples of neurons which signal the second rank and the third. So these are just individual examples, individual neurons. Does population of neurons as a whole represent rank? Let's see. Let's start with the area SEF, supplementary eye field. And what I'm going to show is the overall number of neurons recorded in the area, shown here. The proportion of neurons which signal first rank among the recorded neurons are shown as a red bar, second rank as a green bar, and the third rank as a blue bar. The overall proportion of rank-selective neurons in the given area is shown on the top of the graph. As you can see, in this particular area, majority of neurons signaled rank. That was also true in the other areas we studied. In fact, the rank selectivity was predominant in each of these areas, and a higher number of neurons signaled rank than the neurons which signaled direction, despite the fact that 3 of the areas we were recording from are thought to have motor-related functions, as reflected in the name: supplementary eye field, supplementary motor area, pre-supplementary motor area. So now we know the answer to the first question we posed. In the context of an action sequence task, do neurons exhibit rank selectivity? And the answer is clear, yes. Now we can move on to the second, more interesting questions. Do such neurons represent rank abstractly, regardless of the particular behavioral context? To answer these particular questions, we designed another sequence task, in which, rather than performing ocular movement in the given order, the monkeys were required to select objects in the given order: the red shape first, green shape second, and the blue shape third. The timing of events in this particular task was matched to the timing of the events in the action sequence task. However, the rule governing the target selection was different. The monkey had to select the correct object, first, second, and third, regardless of the object's location in the given phase of the trial. Please note that the locations of the objects are scrambled from one phase of the trial to the next. Therefore, and that was done in each trial. Therefore, there was no consistent relationship between the phase of the trial and the direction of the movement. Well, we found that in the context of this task, in fact, many neurons signal the same rank as they did in the previous task we studied. Let me give you an example. So this is the example of neuron selective for rank 1 in the action sequence task, and we recorded the same neuron in the object sequence task. And we can see that the neural selectivity for the first rank is preserved. We also found the examples of neurons signaling rank 2 in both tasks and rank 3. To quantify the degree of concordance across the two tasks, we performed the following analysis. We first selected all of the neurons which were selective for rank 1 in the action sequence task and displayed neuronal activity in terms of spike per second in each phase of the trial. It looks cartoonish, but it's actually real data, minus the error bar, which you won't be able to see because of the high number of neurons. Now, not surprisingly, these neurons fired the most during the first phase of the trial because that was the basis of our selection. Let's see what do the same 66 neurons do in the second task. They did pretty much the same. That was true for the neurons selected for the second rank and for the third rank. That was the area SEF, but we found that there is a high degree of the concordance across two serial order tasks in rank selective neurons in all of the areas that we studied. We quantified the degree of concordance by computing in each task the measure of selectivity for a given rank a given neuron has. And now we can plot the index, the rank index, which is the measure of selectivity in the object sequence task versus the action sequence task. We can compute the R-squared values and confirm that, in fact, there is a significant and positive correlation between rank indices in two tasks. In some areas more than others, however, there is a still high degree of the concordance in neuronal activity. In other words, whatever the neurons tended to be in the sequence of actions, they tended to do in the sequence of objects. Now we know the answer to the second question that we posed. Do rank-selective neurons exhibit identical rank selectivity in the context of an object sequence task? And the answer seems to be yes. Can we now conclude that the neurons are generalists for rank? Let's vote. Who thinks we can conclude that the neurons are generalists for rank? Okay, 1, 1.5, 2.5. Okay, you're actually correct thinking that, yes, it's a pretty strong evidence. However, we have to consider the possibilities that there are other hidden variables which are common across the tasks. And the first most obvious suspects for the confounding factors of rank selectivity is the passage of time and the expectation of reward. Therefore, we pose the question, can this rank selectivity that we observed in two serial order tasks can be explained by sensitivity to the passage of time or the approach of reward? Let me explain what I mean by that. Notice, as the monkey progresses from one phase of the trial to the next, counting the objects or the actions, the time passes. Therefore, some of the neurons which seemingly signal, let's say, rank 1, in fact, can signal just the beginning of the trial. To control for this possibility, we designed yet another task, which we called long delay task, in which all the monkey had to do was just to wait for the period of time matched to the duration of the typical trial in the serial order task. And at the end, the monkey would receive a reward. We can compute the degree of selectivity, neuronal selectivity for a given rank in the sequential task by computing the rank index. And we can compute identical or analogous measure of selectivity for the passage of time, beginning, middle, and end, in the long delay task. And we can plot the one index, time index, versus the rank index. In this way, we can quantify how much of the variability in neuronal activity we can actually attribute to the neuronal selectivity for passage of time rather than selectivity for rank. We observed that there is a small but significant correlation between rank index and the time index. Now let's leave it for a second and return to the second confounding factor in both sequential tasks, expectation of reward. Indeed, as we progress through the sequence, our expectation of the reward at the end of the sequence grows as we progress through the trial. Therefore, some of the neurons which signal, let's say, rank 3, in fact, can just signal the higher expectation of reward at the end of the trial. To control for this possibility, we designed a task in which we called variable reward task, in which in the beginning of the trial, we signal by the visual cue to the monkey what amount of reward he is going to get after a very short period of wait, which is approximately half of the duration of phase 1. So given the cue in the beginning of the trial, monkey knew at the outset whether he would receive small reward, 1 drop of juice, or big reward, 3 drops of juice. Therefore, we can study neuronal selectivity for the expectation of reward, and by computing the rank index and the reward index, yet again, we can measure how much of the rank index we can attribute to the neuronal selectivity to the growing expectation of reward. We did that, and we found yet smaller than the passage of time, however, still significant correlation between rank index and reward index. At this point, we thought that we may be in trouble because we observed small yet significant correlation in the passage of time task and small yet significant correlation between rank index and reward index. Can these two factors, selectivity for time and selectivity for reward, explain most of the rank selectivity observed in 2 serial order tasks? To answer this question, we did the following analysis. We took the initial correlation between sequential tasks, and then we factored out, by the way of partial correlation, the variability in neuronal signal explained by the neuronal sensitivity to the passage of time and reward. As you can see, only a small proportion of initial correlation can be attributed for codependence of rank signals on time and reward. That was also true in the other areas we studied. So yes, there are clearly some of the neurons which signal both, or only time or only reward. However, the bulk of neurons which signal rank seem to do it regardless of the expectation of time or passage, expectation of reward or passage of time. Therefore, we conclude that correlation between rank indices in the 2 tasks cannot be explained by neuronal sensitivity to the passage of time or approach of reward. We also looked at some of the other confounding factors, but right now we can summarize what we've learned and make certain conclusions. In the context of an action sequence task, we observed the neurons exhibit rank selectivity. They exhibit identical rank selectivity in the context of the object sequence task. Rank selectivity seems to be genuine. It is not an artifact of neuronal sensitivity to the passage of time or the approach of reward. So now, do we think that we found neural correlate of at least certain aspects of the serial order behavior? Who thinks yes? Yeah, a little bit more people. You're more skeptical than the reviewers. Reviewers thought it was pretty convincing. However, and if you have some questions, please feel free to ask them now or at the end of the talk. But finding the neural correlate of a particular behavior doesn't mean proving the causation between the activity of such neurons and our ability to perform a certain behavior. As we all remember, this is the famous comics, the correlation does not equal causation. Well, at this point we can say maybe it is, but we don't quite know for sure. To know for sure, we have to somehow systematically perturb the activity of rank selective neurons in a given area and see if the behavior in the serial order task changes correspondingly. For this set of experiments, we just outright asked directly, does activity of rank selective neurons influence performance in serial order task? And to address this general question, we posed 2 specific questions. The question number 1, does electrical microstimulation of the SAF systematically change the monkey's performance in the serial order task? The microstimulation is the usual way of perturbing or injecting additional current subthresholdly activated the neurons in a given area. So it's pretty well-validated technique of establishing the causal relationship between neuronal activity and a given behavior. We also knew, based on the first part of the project I've described, that the majority of neurons in SEF are rank selective, and among those, the neurons tended to signal later ranks. Therefore, we can form more specific prediction of what we expect by microstimulating this particular area. Specifically, we can ask if it's true that the neurons representing the rank in this area, and most of the neurons represent later rank, does electrical microstimulation of SEF predispose the monkey to select later ranks? To answer this question, we designed the following task, which is analogous to the object sequence task you've seen already. However, now, at the quarter of trial, we injected a very small electrical current into the SEF at the time points before the monkey actually made the decision which object to select. In quarter of trials, we stimulated during phase 2, quarter of trials during phase 3, and we left quarter of trials unstimulated as control. Thus, the monkey's behavior in this particular task, did it change after we did such a procedure? Let's see. So what I'm going to show here on the y-axis is the proportion of saccades, or the eye movements, made by the monkeys toward different objects in each phase of the trial. In non-stimulated trials, the monkey was required to select the first object, and they did, because that's what they were trained to do, but occasionally they make a small proportion of erroneous saccades toward object 2. During the second phase of the trial, again, most of the time they performed the movement correctly. However, when they made a mistake, they split their choices 50-50 between the earlier ranks and later ranks. And during the phase 3, they selected most of the time the correct object, object 3. How did this picture change when we injected electrical current? Let's see. First thing you would notice here that the performance somewhat degraded, seemingly degraded when we did that. The monkey selected the correct object less often. However, if we predisposed to the monkeys to select later ranks, that's what you would predict during the phase 1 and 2. However, the performance during the phase 3 somewhat slightly improved. There is not much room for improvement already. That's all we can see. However, it's very consistent with the ideas that the monkeys are biased towards selection of the later objects. The most informative in this regard are the choices during phase 2. In the non-stimulated trials, monkey, when they were making mistakes, they selected first and third object with equal probability. However, when we electrically stimulated the area SEF, it seemed that we shifted the monkey's predisposition toward the later ranks. Again, this is just the part of this particular experiments I'm describing, but we and the reviewers as well were pretty convinced that actually it matters. If you inject electrical current by somewhat increasing the level of activation of neurons in these areas, it in fact changes the monkey's behavior in the serial order task. So we concluded that the electrical microstimulation in fact changes the performance in the serial order task, and it seems to predispose the monkeys to select later rank, which is consistent with the neuronal activity observed in this area. Let's briefly summarize this part of the talk. We learned that the majority of neurons in the frontal cortical areas is rank selective. Neurons exhibit similar selectivity for rank of an action and the rank of an object. The correspondence between different serial tasks seems to be due to the rank and not other confounding factors such as time and reward. And finally, there is some evidence for a causal relationship between the activity of rank-selective neurons and performance in the serial order tasks. Now, let's pause for a second and move on to the second project that was completed at the Janssen in collaboration with Inscopics. And the aim, the main goal of this project was to investigate the neuronal mechanisms of behavior changed by a drug, specifically by a sleep-inducing drug. And I'm going to motivate the drug selection later on in the talk. The first question we are usually asked in presenting this project is why would the big pharmaceutical company, Johnson & Johnson, would be interested in investigating basic neuronal mechanisms of certain behaviors? Well, the answer is obvious, is that if you don't know what the mechanisms are, you cannot develop the treatments for that. And in fact, current drug discovery is heavily relying on the systematic screening of compounds against isolated biological targets, against isolated receptors, against isolated molecules. The problem with this approach is that such in vitro screening often does not translate into desired effects in preclinical species. Furthermore, in case of psychiatric disorders or mood disorders, it is often not clear at all what the desired effects are, for example, in case of depression. Therefore, the alternative approach was proposed that rather than screening for drugs that influence isolated biological target, is to screen for drugs that reverse pathological network state or promote desired network state in homologous brain regions in preclinical species. To validate this general frame of thinking about drug discovery, we used calcium imaging performed with the miniaturized head-mounted imaging devices. And the example of the device is shown here. And what it basically is, if you look carefully at the diagram, it's the typical benchtop microscope, highly miniaturized and connected to the CMOS camera. It's the same camera chip that you have in your cell phone. The result is that this highly miniaturized camera allows studying activity of large group of neurons in awake behaving rodents. So let me give you an example. So what's hopefully you can see here is the mouse carrying the device is shown to the left, and the activity, the calcium, the fluorescence linked to the changes in intracellular calcium is shown to the right. Don't be alarmed by the frantic movements of the mouse. The videos is sped up. Yeah. So what I hope to convince you by showing this short video fragment is that you can image with this kind of imaging devices. You can image calcium activity in freely moving rodents, in deep brain structures. The example I showed was the neuronal activity in the area CA1 of the hippocampus. It's fairly deep brain structure. It's definitely not on the surface. And we can image the activity of individual neurons. You could see the spots of the flickering in the videos. Those are the individual neurons. And what's not obvious from the movies, but nevertheless true, is that you can image the same neurons over a long period of time, in the order of months. Let me just briefly describe some of the aspects of the calcium imaging procedure, because this technique is still fairly novel. The first thing which is required to accomplish calcium imaging in awake behaving animals is to precisely target the expression of calcium sensor in the area of the interest. And more precisely, you can target the expression of calcium indicator in the given neuronal population. In the example shown here, we used CAMK2 promoter to express calcium indicator in the pyramidal excitatory cells. The second step is to implant the optical part of the microscope over the area of interest by removing the column of tissue and positioning this optical part of the device directly above the area of interest. This is the hippocampus. This is the area CA1. The advantage of using this approach, although it's somewhat labor intensive, is that you can actually directly visualize effect of the different drugs in the neuronal activity in the area of interest. And I'm going to show some examples now. So what I'm going to show, I'm not going to show the mouse, but I'm going to show neuronal activity as we're picking the mouse up, putting the mouse in the anesthesia induction box, and the mouse goes under anesthesia, goes to very deep anesthetically induced sleep. So let's see if you can figure out when the mouse actually went to sleep. When my mouse disappeared. Hold on. Okay, let's try to move it. I'm going to replay it. So replay it again. Here is the normal calcium activity. We're placing the mouse in the box, turning on the anesthesia, and there is no more calcium activity in the hippocampus. So this is the advantage, but how do we quantify what we see in such raw videos? Let me briefly describe a series of steps we do to analyze the neuronal data collected in such settings. First, we can isolate the location of each individual neurons by performing principal component and independent component, principal component analysis and independent component analysis on the motion-corrected movies to identify the location, the source of the highest change in the fluorescence in the raw videos. We can then trace changes in the fluorescence across the trial. Here it's shown just the 10 minutes. By the way, these are the traces of 20 of the neurons taken directly from the movies you just saw. But in the movie I showed about 2 minutes period starting from here and about 30 seconds shown here. So that was the period shown in the movie. And we can trace the changes in the fluorescence and hopefully underlying neuronal activity across the trial by displaying this DF over F changes. We decided to validate this approach by studying one of the commonly prescribed sleep medication, Zolpidem, and study how the activity of neurons in the hippocampus is changed, if it's changed at all, after we administered Zolpidem. Mm-hmm.
So the bursts of light we saw were like clusters of spiking?
Yes, it's bursting. So this is getting into details, but the type of the calcium indicator we used in this particular study was GCAM3. GCAM3 is slow. So usually it's very, very difficult to resolve individual spikes, if possible at all. Sometimes it's not possible, depending on the signal-to-noise ratio. So what we visualized is the burst, indeed, the bursts of actional potential. It's not individual spikes. Yes. Why did we select this particular drug? First of all, the millions, it's close to 20 millions of people per year getting prescribed this particular sleep-inducing medication. Regardless of the fact that it's widely known that there are some severe memory side effects while people are taking Zolpidem, and that you can take directly from FDA Drug Info website. And the key point is here that the next morning you may not remember what you did anything during the night. You have higher chances that there are some other disturbances. But it seems that this drug somehow interferes with our ability to encode the information while we behave under the influence of the drug. And this fact was widely reflected in the popular culture. That was the recent paper in the New Yorker, and this is the series of cartoons you can easily find on the internet. It's widely known. The side effects, memory-related side effects of this particular sleep-inducing drug is widely known. Nevertheless, there are still unanswered questions about Zolpidem effects on neuronal activity, starting with the most basic one. Does Zolpidem change neuronal activity in the hippocampus, the structure which is critically involved in the memory formation? To do that, we image neuronal activity in the two periods. Period one is following the vehicle dose. In vehicle, it's basically a placebo. It's exactly the same solution as the solution containing the drug minus the drug. And then we image neuronal activity following Zolpidem administration. This particular design allows us to compare neuronal activity side by side in two periods of interest. And this particular video, I hope you can see by eye the difference in neuronal activity. Do you see it? So where it seems to be higher neuronal activity, here or here? Let's see, who thinks that it's higher here? Yeah, most of the people, yeah. It's indeed the case, as we can confirm by first plotting the traces of fluorescence across time, indicating the calcium transient which corresponds to the burst of actional potential by a plot, and constructing similarly looking to the electrode recording raster plots of neuronal activity. This is just one animal example, but we can plot it for all of the animals. This is the distribution of calcium event rates in vehicle conditions shown in gray and Zolpidem conditions shown in light gray. And we can see that Zolpidem clearly reduces neuronal activity as compared to the vehicle. That was true in all of the animals we studied. And at this point, we can say, does Zolpidem changes neuronal activity in the hippocampus? And it seems that it does. The related question, and here's the connection to the first part of my talk, how do we know that the Zolpidem is what drives the changes in neuronal activity? As we've shown in the previous examples, there are always a number of confounding factors or contributing factors which changes and influence neuronal activity in no matter settings you're studying neuronal activity. We looked at the set of the parameters which can be covariate with the Zolpidem. And I'm going to just briefly mention two of them. One is the changes in neuronal activity. We asked the question, can the changes, or a decrease more specifically, in neuronal activity be explained by post-Zolpidem changes in locomotor activity? It is known that the people taking Zolpidem behave slower, their motor activity decreases, and that is also true in the animals we administered Zolpidem to. As an example, let me just show you the path of the animal in the arena during the 2 minutes in the post-vehicle period and 2 minutes during post-Zolpidem period. You can see just from this fairly random example that there is a clear decrease in the locomotor activity across two conditions. Can we factor out the contribution of changed locomotion from the observed neuronal effects? Remember that we're recording from the freely behaving animals. Naturally, sometimes the animals are inactive. They stop moving and sit and do something else, or go to sleep, or just locomotorily inactive. Therefore, we can select the time periods which correspond to these periods of locomotor inactivity and study neuronal activity specifically during these time periods. Very briefly, we found that when we just looked specifically at the inactive periods shown in green here, in the vehicle period and the Zolpidem period, there is still very high seemingly Zolpidem-driven changes and diminishing in neuronal activity. Our general answer is that it does not seem to be the case that Zolpidem-induced decrease in neuronal activity is stronger than expected from decreased locomotion. So the final point of this talk, and I'm just going to focus on just one aspect of the experiments that we've done. And the experiments were aimed to trying to isolate the main factor that underlies Zolpidem effects on neuronal activity in the hippocampus. And we looked at a variety of conditions, but what I'm going to show you is the recording of neuronal activity during the non-REM sleep. While non-REM sleep, Zolpidem is a sleep-inducing drug. It is known that it predisposes somewhat animals and humans to increase the sleep duration and reduce the latency to falling to sleep. That's why humans take it. More specifically, Zolpidem increases the non-REM duration. Furthermore, non-REM is a predominant sleep state in all mammals, in us and the mice as well. Therefore, what I'm going to show you is the changes in neuronal activity in the vehicle and Zolpidem conditions, specifically during the non-REM. And to mention briefly how do we know that the mouse is non-REM, we combine the calcium imaging with electrophysiological traditional recording in tracing the EEG and EMG activity, in addition to tracing the animal's locomotion. And therefore, we can isolate and score the time periods when we know that the animal was in non-REM sleep. We can, by coupling the calcium imaging to the traditional EFIS recordings, we can identify and study neuronal activity specifically during the non-REM periods. What do we see? Again, very briefly. What's shown here is the neuronal activity during the baseline periods, and the baseline periods is before we inject anything, before we administer anything, either the vehicle or the drug. And usually we place the mouse in the novel environment to encourage the exploration and movement activity, to kind of drive the neurons to the maximum level. That actually facilitates the later data extraction, data analysis. So you can see that later, when we actually injected either the vehicle or the Zolpidem and studied neuronal activity specifically during the non-REM sleep, even in the vehicle condition, we can see a fairly drastic decrease. However, when you look more specifically, so the first, again, most obvious conclusion from this kind of experiments is that non-REM by itself is associated with low neuronal activity in the hippocampus. This finding is not novel. It's just the confirmation of the previous findings and also validation of the technique. That finding was expected. However, we also found that during non-REM, Zolpidem seems to decrease neuronal activity to the levels below levels expected during physiological non-REM sleep. And this is one of the indications that Zolpidem may suppress neuronal activity in memory-related structure, hippocampus, to below normal physiological level. And our general thinking is, and the project is still ongoing, that that actually explains why people are unable to encode memories while they're acting and behaving under the influence of Zolpidem. So Sorry, I will overrun with time, so I just put the overall conclusions here. The first is that recording and perturbing neuronal activity and correlating this activity with behavior in carefully controlled behavioral tasks is the main tool, still remains the main tool in experimental neuroscience that allows detailed investigation of neuronal mechanisms of behavior. In vivo calcium imaging is a yet even more powerful tool in basic research and drug discovery that can identify neuronal mechanisms of behavior changed by a drug and provide deeper insight into drug efficacy, kinematics, and side effects that other techniques came short to reveal. But the final and the most important point I'm trying to make is that in the multiple electrode and calcium data, when we're collecting massive amounts of data, it's often the case that we have too much information but not enough knowledge. It's just overwhelming, the number of behavioral parameters in the freely behaving animals which can influence neuronal activity and vice versa. Therefore, while powerful tools definitely increase our capabilities of collecting neuronal data, carefully controlled experimental conditions are critical to properly understand the data I've collected in this kind of experiments. That's the end of my talk, and now I can answer some of the questions.
Any questions for Tamara?
Mm-hmm.
I have a question about the first part. You used 3 phases in all your tests.
Yeah.
So I wonder if you used other numbers of phases, say 2 or 4, to check how the curves move.
That is an excellent question. We actually didn't do this experiment, but other people did. And what they found that up to a certain number, so let's look at the task. So here's the, I guess, the example of such neuronal activity. So this type of neuronal selectivity, kind of very sharp, crisp, highly selective neuronal selectivity, it's actually contingent on the number of actions or objects in the sequence. As the number of steps in the sequence increases, it seems to be, the selectivity seems to be spreading out and leaking into the neighboring ranks. That's kind of one of the reasons we may find the long tasks much more difficult than the short tasks. However, again, there is still a number of neurons which do preserve this kind of very abstract, clear rank selectivity in different tasks. So it seems to be, yes, it is contingent on that. However, there is still a number of neurons, and we expect that in humans such number would be higher than in the monkeys, that signal the exact ordinal position in the sequence, regardless of the number of elements of the sequence. And just a side note from information perspective, strictly speaking, it's not necessary to have this kind of sharp, crisp, abstract rank selectivity which generalize across several tasks. However, computationally, it makes things easier. If you're faced with the task with a longer sequence, you can hierarchically subdivide it into the elements and then associate each element in the sequence into with the neuron representing given position in the sequence. That makes sequence learning much more efficient, and there is some modeling studies which actually confirm that. So the presence of such neurons is well justified from the, again, computational perspective, from the point of view of representation of the rank. The interesting point of that is that, strictly speaking, this kind of sharp neuronal selectivity is not necessary. Having widespread neuronal tuning, for example, when you have the sequence of 12 movements, having the neurons representing the widespread middle of the sequence, right, let's say the object 6 to 8, and having other neurons, it's kind of akin to the coding of the colors, when no single neuron codes the color absolutely in terms of the wavelengths. However, the large number of neurons with non-sharp, widely spread tuning are capable to represent as a population each given rank or each given color fairly specifically. Does it answer the question? Okay.
Other questions?
Mm-hmm.
So I have a question also about the first part, the stimulation experiments. So
right.
To a non-neuroscientist like me, it seems kind of surprising that you're recording from regions of the brain that have hundreds of thousands or millions of neurons, and this very sparse sample of a few hundred neurons could elicit such a gross change in behavior. Were you surprised at all by that? Or?
Actually, we weren't surprised in particular, because it's been previous work, and most notably, there is a lab at Stanford at the direction of Bill Newsom, who've done monkeys research for years. And what they found is that they were studying neuronal activity in the area MT. This area is thought to be sensitive and underlying the ability to appreciate motion in the different directions. And they found that the carefully subthresholdly stimulating neurons in this area, if you look exactly at what you're trying to find, right, if you're trying to stay away from all the other contributing factors, in fact, even stimulating a small group of neurons can have measurable changes in the overall monkey's behavior. And considering that the rank selectivity in this particular area is much more predominant than any given direction selectivity in area MT, we sort of expected that it would be visible in the behavioral data. It's very surprising. Actually, if you Google electrical microstimulation, you would find the plethora of movies of people using different focusing devices by injecting the subthresholdly stimulating the different brain regions and so forth. This is quite dangerous, frankly speaking, especially there are some recipes how to get the Radio Shack elements and make this kind of things. However, yes, it does indicate that there is something about injecting small electrical currents in the given area of the brain which predictably changes the behavioral in the different set of situations. And that's very consistent with decades of the neurophys research in the monkey neurophysiology.
All right, let's thank Dr. Berdyyeva.